Chapter 32 is our first chapter in our animal unit, and it's covering the internal organization of animals and regulation. So we'll talk about homeostasis, animal tissues, a big picture on organ systems, a tiny bit on endocrine and nervous system. We'll get to nervous system in more detail later, and we'll talk about feedback cycles and homeostasis and thermal regulation and osmoregulation and excretory systems. So again, this is kind of a big lump, and maybe if I had written this book, I would have split it up a little bit more than this, but here we go. Homeostasis is the word to describe a kind of steady state or internal balance in an organism. So the outside environment can change, but the organism's internal environment stays in a relatively stable state. So there are some changes to your internal environment, like your pH can have slight variations, or your body temperature can have slight variations. So this moderates change, it doesn't entirely eliminate change. A good example of this, or a good thing to think about to represent this, is if we moderate the room temperature with the help of a thermostat and a furnace, or a thermostat and an air conditioner. So as the temperature changes one way, the thermostat is either turned on or off, and then the room temperature will go back to that set point. So there's different homeostatic control systems, and in a homeostatic control system, we have a set point, which is a particular value that's the normal range. And again, the set point is usually relatively stable, and fluctuations above or below that set point serve as a stimulus, and that stimulus is detected by a sensor, and it leads to a response, and that response returns the variable back to the set point. So just like the room temperature example, if it gets too hot, the air conditioner flips on, and the room gets cooler, and then the air conditioner turns off. So feedback is a fundamental feature of homeostasis, and this is how we can really minimize major disturbances. Feedback can occur at the organ level, the cellular level, or even the molecular level. The thermostat example we talked about is a negative feedback cycle, because the variable that's being regulated brings about a response that moves the variable in the opposite direction. So the room gets too hot, the AC turns on, cools the room off, and now we're back to the set point. Negative feedback cycles prevent homeostatic responses from overcompensating or going too far. There's also positive feedback cycles. Positive feedback cycles accelerate a process. They go in the same direction as the initial stimulus. If we think about a domino knocking over the next domino, knocking over the next domino, knocking over the next one, and so on and so forth until all the dominoes are knocked over, that's a good example of positive feedback. So positive feedback is called positive feedback because it goes in the same direction as that initial change and it adds to it, while negative feedback goes in the opposite direction of that original stimulus. So let's look at an example of negative feedback. Maintaining your blood glucose levels or your blood sugar levels. I feel like most people have heard of insulin before, so we'll start with insulin. If you start up here with high blood sugar, the pancreas releases insulin, which tells the tissue cells to take in glucose, and then those cells taking in glucose from the blood lowers the blood sugar. Insulin also tells the liver to convert glucose to glycogen, and so now we have less glucose, so that lowers blood sugar. However, there's another end to this feedback cycle. If you have too low of blood sugar, the pancreas is stimulated to release glucagon, which tells the liver to convert glycogen into glucose, so break down glycogen to release glucose, and that increases blood sugar. So both of those are examples of negative feedback. An example of positive feedback, this is really pretty simplified because obviously delivering a baby, there's more to it than this, but if we look at kind of the simplified picture here just to sort of understand positive feedback, if we start here at number 1, as the uterus contracts, it causes the cervix to stretch more, and then you get a signal from the stretched cervix to the brain, and then the brain triggers the pituitary gland to release hormones that travel to the uterus through the bloodstream, where they produce stronger contractions, which causes more cervical stretching, and so on and so forth, all the way until the delivery is complete. So that's positive feedback. So take a look at this example and let me know what kind of feedback it is. Is it positive or negative? If you need to pause it, go ahead. So in this example, we have our homeostasis or balanced state here of normal body temperature. This person is cold, so receptors in their skin and their brain are temperature sensitive, and they're like, oh my gosh, it's cold. So they send that information along the afferent pathway to the control center in the brain. The control center in your brain is like, oh, we're cold, we better do something about it. So they send that message along the efferent pathway. Quite honestly, I'm going to say efferent from now on, even though that's not the way you say it correctly. But I think it's easier for students to tell the difference between afferent with the A and efferent if I say efferent because of the E. So then the efferent pathway or efferent pathway sends a message to the skeletal muscles that cause the person to shiver, and then that shivering raises the body temperature back to balance. On the other side here, we have basically the opposite if the person's too hot, and then they sweat to cool down. So is that positive or negative feedback? That's going to be negative feedback because if we're too cold, we get warmer, so it's going in the opposite direction. If we're too hot, we cool down, so going in the opposite direction to maintain homeostasis. So what kind of feedback is this? Again, pause it if you want. So in this feedback cycle, you have, and again, this is sort of simplified just for the sake of understanding feedback cycles more than understanding blood clotting, but we have a tear in a vessel wall here, and that initiates a feedback cycle where platelets adhere to that tear and release chemicals that call more platelets to the area, that call more platelets to the area, and so on and so forth until we get a platelet plug formed. So that's going to be a positive feedback cycle. So anatomy and physiology are related to each other. They go together. Anatomy is the fancy word for structure. Physiology is the fancy word for function. And a structure is related to a function, or the function is related to the structure. Multicellular organisms can really specialize their bodies because they've got these different cell types, and they can devote certain cells to certain activities. And animal bodies have different kind of layers of organization. So let's look at some terms for organization in the animal. I think you should pretty well be able to do this. I didn't do the best job taking the labels off of the picture. So in the first one here, we've got a muscle cell, a neuron, epithelial cell, and connective cell. So those are all going to be at the cellular level, pretty obviously. And then when we have many muscle cells working together, we get muscle tissue. Or many neurons working together, we get nervous tissue, and so on and so forth there. So that's going to be the tissue level. And then when we have different tissue types working together in the bladder, that's an organ. And then here we have the bladder, the ureter, and the kidneys, and the urethra. So that makes up the urinary system, which is an organ system. And then this last one here, we have the entire chimpanzee there, which is the organism. So if we look at animal tissues, again, tissues are specialized cells working together. Animals have muscle tissue, nervous tissue, epithelial tissue, and connective tissue. Muscles contract and generate force. Nervous tissue initiates and conducts electrical signals to send messages. Epithelial tissue is covering and lining tissue, and then connective tissue is a big group. Anybody who takes anatomy and physiology, probably anatomy, I guess, would know that there's lots of different kinds of connective tissue. For example, for different tissue types here, we'll start with connective tissue, like you've got elastic connective tissue, you've got blood as a connective tissue, bone as a connective tissue, you've got dense regular or dense irregular connective tissue, kind of on and on there. There's nervous tissue in the brain of this wolf. They're showing you skeletal muscle tissue here, and then epithelial tissue here lining the intestine. As far as organ systems go, we're going to hit specific organ systems in more detail later, and if we were actually in class together, I would ask you, or the people around you, what the function of different organ systems were, and we would kind of chat about it, but we'll do kind of the best we can to replicate that here. So if we start with the integumentary system, that's your skin. So it's going to cover your body. It's going to protect you from pathogens and from, abrasions from the environment, that sort of thing. We've got the skeletal system, which is important for protection and support. It also functions for storing minerals. Muscular system is important for movement. Excretory system helps us maintain our fluid balance and salt balance. Nervous system sends and receives electrical impulses. Endocrine system, that's supposed to be the thyroid gland. It's not like a bumpy butterfly. So endocrine is hormones. You've got your circulatory system, which pumps blood. Reproductive system, I think, is pretty straightforward. Produces some hormones, and then also helps produce more of a particular organism, right? The immune system and lymphatic system are kind of lumped together, but really they have separate functions. Lymphatic system helps return fluid to, like extracellular fluid, to our blood, and then immune system helps fight infection. And they're kind of lumped together because there's an overlap between lymphatic and immune system, which we'll get into more later. And then respiratory system helps do gas exchange with the environment, and then digestive system helps you break down large molecules into smaller molecules and helps you absorb those molecules. In order for your tissues and organs to work together, they need to be able to send messages to each other and receive messages from each other. So that's kind of the role of the endocrine system and the nervous system. The endocrine system releases molecules into the bloodstream, and that can reach locations throughout the body. The nervous system is where we have neurons that transmit signals along a specific pathway, and that connects specific locations to the body. Again, the endocrine system releases hormones, which are these signaling molecules that's sent out by the endocrine system. They can have an effect in a single location or throughout the body, but only cells that have receptors for a particular hormone can respond to that hormone. And the endocrine system is really good for coordinating gradual changes that affect the entire body. The nervous system is better at or it's well suited for directing immediate and rapid responses to the environment. It sends signals called nerve impulses along the axon of neurons, and the nervous system can signal other neurons or muscle cells or endocrine cells or even exocrine cells. So all these cell types can receive nerve impulses and respond to them. As far as glands go, the endocrine cells are often grouped in ductless organs called endocrine glands, and endocrine glands and cells secrete hormones directly into their surrounding fluid, from which the hormones enter the bloodstream. For example, antidiuretic hormone is a hormone. And then exocrine glands have ducts, and that carries enzymes or secreted substances into body cavities or onto body surfaces, so something like mucus would be produced by an exocrine gland. So here's just some pictures of some glands here. I don't expect that you memorize these, but we're showing you kind of the endocrine system in general. And not that memorizing them is impossible or unreasonable, but unfortunately we only have 2 units on animals and we just don't have a lot of time. So there's certain hormones that I do want you to memorize, and we'll get to those later. So if we look at a simple endocrine pathway here, the pH of your stomach is low. Your stomach contents empty into the small intestine. The first part of the small intestine is called the duodenum. The duodenum and the small intestine in general don't have as much protection as the stomach does, so they need to correct that low pH. And there's more to that as well, but that's kind of the big picture. So our stimulus here is a low pH in the duodenum, so low pH in that first part of the small intestine. That causes some cells of the duodenum to release a hormone called secretin. That hormone circulates in the blood. It reaches target cells in the pancreas, which release bicarbonate. Bicarbonate is alkaline or basic, so that will raise the pH back to a more neutral level there. So hormones can either be water soluble or lipid soluble. If a hormone is water soluble, it can't make it through the cell membrane because remember the cell membrane is a phospholipid bilayer largely. So instead of crossing the cell membrane, it binds to cell surface receptors, and that triggers events leading to the cellular response. Remember, in the cellular response we have the ligand, so the hormone binds to the receptor, and then we have signal transduction, and that signal transduction pathway often has multiple steps, but then we get a cellular response. If we have a lipid-soluble hormone, lipid-soluble hormones can make it through that phospholipid bilayer, so they're going to bind to receptors inside of the cell. And when the receptor and the hormone are bound together, they typically are going to move to the nucleus and alter transcription of particular genes. So remember, if we transcribe a gene, we make the RNA, which means we make the protein. So basically that's saying they turn on certain genes. Many hormones have more than one response in an organism. For example, epinephrine is secreted by the adrenal glands, and it raises blood glucose levels, it increases blood flow to muscles, and it decreases the blood flow to the digestive system. Target cells really vary in their response to hormone because they differ in receptor types and the kind of molecules that they produce in response to the hormone or the ligand. So here's a couple examples of cells and receptors and cellular responses. So if we look at cells that have the same receptors but different intracellular proteins, so here we have liver cells and here we have smooth muscle cells. They both have beta receptors for epinephrine, and in the liver cell it causes the glycogen to be broken down to release glucose to increase blood glucose levels. In the smooth muscle cells, it causes the muscle cell to relax. And I should say smooth muscle cells with beta receptors. So epinephrine binds to the beta receptor, that causes the cell to relax. So that could dilate or make the diameter of a blood vessel larger to increase blood flow to skeletal muscles. Interestingly though, epinephrine constricts blood vessels going into the intestines, but the reason this happens is because smooth muscle cells in the wall of the blood vessels that supply the intestines have alpha receptors, so same hormone, same cell type, different receptor, different response. The smooth muscle going to the intestines contracts, the smooth muscle going to your skeletal muscles relaxes. So a hormone's response depends on the receptor and the cell type. Over the course of evolution, the functions of a given hormone have often diverged between species. For example, we have thyroid hormone, frogs have thyroid hormone, but in frogs, the thyroid hormone plays a role in stimulating reabsorption of the tadpole's tail during metamorphosis. That's not really a function that we have with thyroid hormone. Let's review then. Number 4, which of these is an exocrine gland? So remember, exocrine secretes onto surfaces, and endocrine goes into the bloodstream. So the first one here in the picture is exocrine. The second one is endocrine because the hormones are going into the blood in the second one. Number 5, the insulin receptor is transmembrane. Would you expect insulin to be water or lipid soluble? So if you look at the word transmembrane, that means it's across the membrane. So that means the insulin itself isn't making it across the membrane. So would you expect it to be soluble in water or lipids? If we have to have this membrane receptor, that means it's going to be water soluble, because if it was lipid based, it could just go right across the cell membrane on its own. So insulin is water soluble. As far as regulating and conforming goes, now we're kind of talking about how animals manage their internal environment. They can do this either by using internal mechanisms to control their internal changes despite external fluctuation. That would be a regulator, like they're regulating their internal environment. Or if they're a conformer, they allow their internal condition to change in accordance with the external changes. And some animals regulate some factors and conform in others. For example, a fish may conform to surrounding temperature in the water, but it regulates solute concentrations in its blood and interstitial fluid. The interstitial fluid is just the between the cells fluid. So here we've got a graph that's showing us ambient temperature in the environment and body temperature of the organism, and then we're looking at a river otter, which is a temperature regulator, so its body temperature is relatively stable. And then here we have a largemouth bass, which is a temperature conformer, and you can see as the environmental temperature increases, so does the fish's body temperature there. Thermoregulation is the process by which animals maintain their body temperatures within a normal range. An endothermic animal is able to generate heat through their metabolism, and they can maintain their body temperature in the face of large fluctuations in environmental temperatures. Not to say that they will be able to maintain it in all environmental conditions, right? People and animals can die of hyper or hypothermia, but we can maintain a relatively stable internal temperature. Ectothermic animals gain heat from external sources, and ectotherms would include things like most invertebrates and fishes and amphibians, and then a non-avian reptile, which would be a regular reptile, not a bird. Oftentimes, ectotherms regulate their temperature by behavioral means, and generally they do not consume as much food as endotherms because their heat sources are largely environmental. That's not to say endotherms never regulate their temperature behaviorally either, right? If it's hot, do you ever stand under a tree in the shade, right? If it's too hot, you go in the shade. That's behavioral regulation. But that's not the only thing your body is capable of doing to help maintain temperature. There are 4 heat exchange mechanisms, and if you take a physics class, you'll probably talk about these in more detail. We're going to talk about them with relation to animals and exchanging heat with their environment, but this works for other things as well. So radiation is emission of electromagnetic waves by the surface of objects, and the rate is determined by the temperature of the radiating surface. Evaporation is when the heat that's required to drive the process of water vaporization from a body's surface is conducted from the surface. It cools the animal. But I think you know what evaporation is, right? You sweat, the sweat evaporates, you cool down. Convection is the transfer of heat by the movement of air or water next to the body. So if you've ever sat near a fan, or if you've seen an elephant wave its ears or a bat flap its wings to cool down, that's convection. Or if you have a convection oven, it has a fan in it that blows the air in the oven around. Like part of the oven is heating with radiation, but then the convection oven also has this fan that blows the hot air around, and it's supposed to cook your food more evenly. Conduction is when you gain or lose heat with direct contact with cooler or warmer substances. If you think about getting into a body of water, right, and the water is cold, that'll cool you. Also, if you always think of like laying on a hot rock in the sun, but then that's really 2 things, because you have radiation from the sun and then you have conduction from the hot rock. Or if you've ever been camping and you've laid on the ground at night without a camping pad and gotten really cold, that's because the ground is taking the temperature from, or taking the heat from your body and lowering your body temperature. So heat is always going to be transferred from an object of higher temperature to one of lower temperature. Organisms have circulatory adaptations for thermoregulation as well. Animals can actually alter the flow of blood and hence heat between their core and their surface of their body. You already kind of know this. You haven't really had the chance to meet me per se, or maybe you can't see me right now, but I'm pretty pale, and if I run, my face flushes, right? Or even if I get embarrassed, my face flushes. because I have vasodilation on the surface blood vessels. So that widens the diameter of those surface blood vessels, and then I have more blood flow to my skin, and that promotes heat loss. Hopefully you've never seen it, but if you've ever seen somebody who's very, very cold, like on the verge of or having hypothermia, they appear kind of like bluish, hopefully not too blue, but that's because they've done vasoconstriction in the blood vessels on the surface of their skin, and so their body's doing that to try and reduce heat loss there. Some organisms use a countercurrent heat exchange mechanism. A countercurrent mechanism is going to have fluids running in opposite directions, and in this case, since it's a countercurrent heat exchanger, heat is going to be exchanged from these fluids that are in like parallel tubes running in opposite directions. So there's a couple pictures here of birds and bird feet and arteries and veins and the legs of birds. So birds do maintain their body temperature, but if you're an aquatic bird and you're standing in cold water, the water is very cold, but you don't want to cool the whole entire bird down. So as the blood goes down the bird's leg, it's running parallel to the blood that's returning to the bird's heart. The blood that's returning to the bird's heart is warmed by the blood coming from the heart going to the bottom of the foot there. So it's a countercurrent heat exchange. This other picture is showing you the same sort of idea here, where the artery is warming the blood in the vein as the vein goes back up to the heart in the bird there. And here you can see a cross section where the artery is surrounded by veins in this case. So endotherms, like birds and mammals, vary their insulation to acclimate to different seasonal temperature changes, and ectotherms can often make adjustments at the cellular level. Some ectotherms even can produce antifreeze compounds to prevent ice from forming in their cells. There's a short horn skullpin down here that does that. And thermoregulation in mammals is controlled by the hypothalamus, where there's a group of nerve cells that function as the thermostat. The hypothalamus triggers heat loss or heat gaining mechanisms, and fever is the result of a change to the set point for a biological thermostat. So really, like, we all have a temperature, right? No matter what, we all have a temperature. Even if you're dead, you have a temperature, but a fever is an abnormally high body temperature. This is just kind of a little pet peeve thing, but be careful on that one. So here's an example of a feedback cycle regulating body temperature. We've already kind of talked about this before when we were talking specifically about feedback cycles, but here if we have our normal body temperature, if the body temperature increases, the hypothalamus detects that causes blood vessels in the skin to dilate, causes the organism to sweat, and that decreases the body temperature. If we get too cold, hypothalamus says that leads us to shiver, causes the blood vessels in our skin to constrict, and that will cause the body temperature to increase back to the set point. So let's review. Is this organism likely to be an endotherm or an ectotherm? It is an ectotherm. And then let's label 1 through 4 as either radiation, convection, evaporation, or conduction. So go ahead and pause it if you want to. So the first one here, we've got the sun warming up the lizard from electromagnetic rays, so that's going to be radiation. I think number 2 and 3 are a little bit tricky, but 2 is showing you coming from the iguana's nose there, and I think that one's probably the trickiest, but that is showing heat loss through evaporation because the moist air leaving the iguana is coming through its nose and cooling the iguana. They kind of pant a little bit, not the same as a dog, but similar. Number 3 is like a breeze blowing across the back of the iguana, so that's going to be convection, and then here the iguana is on the rock, so that would be heat exchange through conduction there. Number 8, we have a picture of a whale's tongue where warm blood is cooled as it goes to the tip of the tongue, and cool blood is warmed as it goes back to the whale's heart. So what's that? We have two fluids that are parallel to each other, running in opposite directions. Two fluids and tubes running parallel to each other but in opposite directions. So that's countercurrent heat exchange mechanism. So then this is like kind of another, if I was writing the book I might make this a different chapter, but it kind of goes in this chapter because we're still regulating. But if we were going to just go with, oh, we're still regulating, we might as well put everything in the animal body in this chapter, right? Okay, now tell me how you really feel, Miss G. That's how I feel. So, osmoregulation is the general term for the process by which animals control solute concentrations in their interstitial fluid and balance water loss and water gain. Osmolarity is the solute concentration of a solution, and that determines the movement of water across a selectively permeable membrane. So if two solutions are isoosmotic, the movement of water is equal in both directions, and if two solutions differ in osmolarity, the net flow of water is from the hypoosmotic to the hyperosmotic solution. But that should be pretty much a review there. So organisms can either be osmoconformers or osmoregulators. Osmoconformers are going to be isoosmotic with their surroundings and do not regulate their osmolarity, whereas osmoregulators are going to have to spend energy to control their water uptake and water loss in either hyperosmotic or hypoosmotic environments. Organisms have different challenges based on the environment they live in. I think that's pretty obvious if you pause and think. So sometimes I think people get so stuck on memorizing in this part of the unit that they don't just take a second and think. A marine fish lives in a saltwater environment. What's its big challenge? Salt, right? So marine fish drink a large amount of seawater to balance water loss and excrete salt through their gills and kidneys. What's the freshwater fish's problem? Lots of water, not much salt. So a freshwater fish is going to drink almost no water and replenish salts through eating. Some also replenish salts by uptake across the gills. And then a land animal, what's its biggest problem with water? Getting it, right? Not drying out. So land animals have mechanisms to prevent dehydration. Most of us have body coverings that help reduce water loss, and also we drink water and eat moist foods, and we produce water metabolically. When we say we produce water metabolically, do you remember hydrolysis and dehydration synthesis? Dehydration synthesis is where you join monomers together by taking out an H from one monomer and OH from the other, so you take out a water, so that's metabolic water. So here's just a picture of osmoregulation in a marine fish. Again, their challenge is going to be too much salt, not enough water, so they're going to have to excrete salt from their bodies and try and keep as much water as they can. They're also going to lose water through their gill surfaces and other parts of their body, so their goal is to keep water and get rid of excess salt. If we're talking about a freshwater fish, they have the opposite problem. They have too much water. Water can even rush in through their gills and other parts of their body. So they're going to try to keep salt and excrete excess water there. And I don't know, sometimes biology diagrams kill me because you're like, you can tell by the size of the arrow how much water it's excreting, right? There you go. So then a land animal has different challenges as well. This isn't from our book, but I think that it's worth showing you. So ingesting food introduces salt and water. Breathing is going to cause you to lose water. Your metabolism can both use and produce water. When you eliminate waste, you're going to lose both salt and water. And then your body temperature regulation can cause you to lose water as well if you sweat and that water is lost through evaporation there. So those are kind of the challenges of a land animal. So different animals produce different nitrogenous wastes. Nitrogenous wastes are going to be produced by all kinds of animals though, because they're a product of protein and nucleic acid breakdown. Nitrogenous wastes are a bad deal because they're toxic at high concentrations. There's 3 forms. You could argue and you could be like, well, it's really kind of 4, but we lump ammonia and ammonium together, and then we have urea and uric acid. and the type and quantity of an animal's waste products are going to greatly affect their water balance, and there's several mechanisms that have evolved for excretion, which is the process that rids the body of nitrogenous metabolites and other metabolic waste products. So if we look at ammonia, urea, and uric acid, so ammonia is really the most toxic to organisms, but the organism doesn't have to spend any ATP to convert it to a less toxic product, but it does require lots of water for excretion. So if you've ever had a fish tank before, you've either monitored ammonia levels or done water changes and things like that to try and control it in your fish tank. Urea is what we use. Urea is less toxic than ammonia. It requires less water for excretion, but it does require ATP to convert ammonia to urea. Uric acid is even less toxic than urea and ammonia. However, it's energetically expensive to make from ammonia, and things that have like a kind of semi-solid nitrogenous waste product like a bird or an iguana or some insects would produce uric acid. You've all seen bird poop, right? If not, step outside. This is just another kind of diagram showing you breaking down nitrogen-containing macromolecules and producing nitrogenous wastes, and showing you like different kinds of animals use different kinds of nitrogenous wastes. Most fish are going to use ammonia, mammals, most amphibians, and some fish use urea, and then birds and many other reptiles, and insects and land snails are going to use uric acid. If we look at excretory processes, generally osmoregulation and metabolic waste disposal relies on having transport epithelia. Remember, epithelia are those covering and lining tissues. So transport epithelia are cells that are specialized for moving solutes and a controlled amount in a specific direction. And many animal species produce a fluid waste by refining something called a filtrate, which is derived from body fluids. And most excretory systems involve filtration, reabsorption, secretion, and excretion. Filtration is filtering body fluids. Reabsorption reclaims valuable solutes so that we don't lose them. Secretion is when an organism adds solutes to the filtrate. And excretion is when that filtrate is ultimately going to leave the organism's body. So here's just a kind of generalized diagram here to show you those steps. So here we have a blood vessel, a tuft of capillaries, and then another capillary here. And so at this first step we have filtration, where the tuft of capillaries is literally filtering out the blood, and we get something called the filtrate, which is liquid and solutes here in this excretory tubule. The body's going to reclaim some of those solutes in reabsorption. For example, like you would want to reclaim glucose. And then secretion, the body spends energy to get rid of some solutes, and then you have your final urine product that is excreted from the body. So if we talk specifics about different organisms, flatworms have something called protonephridia. If we think of the word proto, I think of prototype, so it's kind of like an early excretory organ. It's a network of tubules connected to external openings in the body. So here, if you can see in this picture, there's an opening in the body wall, there's these tubules, and then the smallest branches of the network are capped by this cellular unit, which is called a flame bulb. It kind of looks like a flame in like a lantern, sort of. It kind of whips around with these cilia. And this structure produces a dilute urine that is then just emptied into the external environment through these openings in the body wall. So that's the protonephridia. The Malpighian tubules are found in insects and other terrestrial arthropods. They help to remove nitrogenous waste from the hemolymph. The hemolymph is the extracellular fluid in organisms with an open circulatory system. So they're removing nitrogenous waste without a filtration step. Instead, they use secretion, and these tubes or tubules are attached to the gut and empty directly into the hindgut, which is then going to leave the organism with feces. So we have kidneys. So invertebrates and some other chordates, the kidney functions in both osmoregulation and excretion. The kidney is made up of a series of tubules that are arranged in an organized kind of array, and also there's a network of capillaries involved as well. The excretory system includes ducts and other structures that carry urine from the tubules of the kidney out of the body. If we look at the structure of the mammalian excretory system, you've got the kidney, which has a renal cortex, which is largely involved in filtration, and then the middle portion here is the renal medulla, which is where the filtrate is going to be concentrated. There's also a ureter, which takes the urine from the kidney to the bladder. Then you have the urinary bladder, and then you have the urethra, where the urine actually exits the body. The nephron is really the functional unit of the kidney, and it's hard to get a good diagram because there's so many in the kidney. They would all be overlapping and kind of it would be very hard to see. So there's as many as several million nephrons in each kidney. The nephron has a renal corpuscle, which forms the filtrate. It consists of the Bowman's capsule and the glomerulus, and then there's the tubule, which performs secretion and reabsorption. We break the tubule down into the proximal tubule, the loop of Henle, which is sometimes called the nephron loop, and the distal tubule. Sometimes you might hear a proximal tubule called the proximal convoluted tubule, and distal tubule called the distal convoluted tubule, because they're twisty and convoluted. The tubule then empties into the collecting duct. So, the whole thing's very twisty and kind of tricky to see, so deep breath. We're going to go over the nephron, and then we'll go over it more later. But to kind of orientate you here, here we have the collecting duct, which actually isn't part of the nephron, but many nephrons attach to the collecting duct there. And we have this arteriole here, which is a very small diameter artery. There's this tuft of capillaries called the glomerulus, and the blood is filtered and produces the filtrate in the Bowman's capsule. So the Bowman's capsule and the glomerulus together make up the renal corpuscle, and then the filtrate moves to the the proximal convoluted tubule. Proximal and distal are anatomical terms. Proximal means near point of origin, distal means away from the point of origin. So the proximal tubule is close to the renal corpuscle. So it's proximal tubule first, and then down one side of the loop of Henle, up the other side of the loop of Henle, or nephron loop, and then to the distal convoluted tubule, and then connecting to the collecting duct there. So if we look at the renal corpuscle, it is made up of the Bowman's capsule, or the glomerular capsule, and the glomerulus, which is this tuft of capillaries here. So the glomerulus has these cells that cover the capillaries that are called podocytes. They have pseudopods, kind of. They're little foot-like extensions is what the word means, and they form filtration slits, which are a physical barrier, and the blood goes in one arteriole here, and then it's filtered out through these pores in the glomerular capillary, and then blood leaves this way. And the way we get it to filter out is we actually have a larger diameter vessel going in and a smaller diameter vessel going out, and so the pressure pushes the solutes and water out of those cells. And really we could go into it in a lot more detail, but we're kind of limited in the scope of things in this particular class. But the glomerular filtrate is what's formed here. Eventually that will be concentrated into urine, but at this point it's not urine yet, it is filtrate. Typically proteins and blood cells are going to remain in the plasma, although sometimes they can make it through, and sometimes that can be a warning of other issues or damage to the kidney. Sometimes it's not, though. It just sort of depends. And again, the Bowman's capsule, or glomerular capsule here, encloses the Bowman's space. So the tubule then is continuous with the Bowman's capsule. So here we have our Bowman's capsule. Here is our near the point of origin tubule, so that's the proximal tubule. Then you have the loop of Henle or the nephron loop, and then you have the distal tubule, and it connects to the collecting duct, which is not part of the tubule. And then you also have paratubular capillaries that are, para means around or near, so they are around or near the tubules. So they're near the cortex and the medulla junction. And you also have vasa recta capillaries here around the nephron loop or loop of Henle. We're going to briefly summarize what happens at each of the parts of the tubule, but again, this is not really the whole story. And if you take physiology, hopefully you get some more information there, maybe anatomy as well. So in the proximal tubule, the main things that are going to happen is the body is going to reabsorb ions and water and nutrients that it wants to keep. However, there's a limit to how many they can reabsorb because there's a limited number of transport proteins. Some things are passively transported and some things are going to be actively transported where that would require ATP. And then some toxic materials are actively secreted into the filtrate here as well. On the descending loop of Henle or descending nephron loop, you have reabsorption of water through channels called aquaporons. These are proteins in the membranes, and movement is going to occur due to high osmolarity of the interstitial fluid, which is hyperosmotic to the filtrate. The filtrate is going to become increasingly concentrated as it journeys down the descending limb. On the ascending limb of the nephron loop or loop of Henle, the transport epithelial tissue here lacks water channels, so salt but not water is able to move from the tubule into the interstitial fluid, and the filtrate is going to become increasingly dilute as it moves up towards the cortex. On the distal tubule, that's important for regulating potassium and sodium chloride concentration of the body fluids. Also, it's important for pH regulation. The collecting duct then, which is not part of the tubule but the tubule is connected to, is going to carry filtrate to the renal pelvis, and it also determines the concentration of the urine by regulation of the permeability and transport across its epithelium. So if we look at it kind of all together here, here we have the glomerulus and the Bowman's capsule, proximal tubule, descending limb of loop of Henle, ascending limb, distal tubule, and collecting duct. The nephron loop here, or loop of Henle, is important for building the concentration gradient from the cortex to the inner medulla. The collecting duct is important for the final concentration of the filtrate into urine, and then the proximal and distal tubules are important for reabsorption of some nutrients, and then also secretion of certain items as well. So mammals need to be able to concentrate water because they're living in a terrestrial environment, and variations in nephron structure and function make some organisms better suited to living in drier environments. So the longer of a nephron loop you have, or the longer of a loop of Henle you have, you're going to be able to produce a more hypoosmotic urine or a more concentrated urine. The loop of Henle and the surrounding capillaries act as a type of countercurrent system. Remember, we had a countercurrent heat exchanger. We also have this countercurrent system here as well. And this system involves active transport, so it does require energy. And since we're using active transport here, we call it a countercurrent multiplier system. So which of the following requires the most water to excrete? Urea, uric acid, or ammonia and ammonium? That one's going to be C, ammonia and ammonium. And then two processes necessary for animal life that could upset water or salt balance. That should be pretty, there's a whole bunch you could name there. So breathing upsets water balance, eating upsets water balance, sweating. It's a pretty long list, but there's a couple for you. Metabolic processes, right? So really the kidney is regulated by a combination of nervous and hormonal inputs. And these inputs contribute to homeostasis for blood pressure and volume as well. So we'll go over antidiuretic hormone and the renin-angiotensin-aldosterone system. Antidiuretic hormone makes the collecting duct epithelium temporarily more permeable to water. So if we think of the word antidiuretic, what's a diuretic make you do? Makes you pee. What's anti mean? Not. So antidiuretic hormone is the not pee hormone. So if the collecting duct epithelium is more permeable to water, that means less water will be lost as urine because you're going to reabsorb more water. So when you have an increase in blood osmolarity above a set point, so more solutes in your blood, that triggers the release of antidiuretic hormone, which helps you conserve water. If you have decreased osmolarity, that causes a drop in antidiuretic hormone secretion and a corresponding decrease in the permeability of the collecting ducts. So you pee more. So if we take a look at it here, if we have our normal blood osmolarity and then it increases, like after you sweat profusely, you've lost water but still have solutes, and you lose some solutes too, but you get the idea. So you have osmoreceptors in your hypothalamus that trigger the release of antidiuretic hormone. That makes the collecting duct more permeable to water, so you lose less water. You also become thirsty and you drink more water, so that helps restore your normal blood osmolarity. So let's go ahead and label the excretory processes on the diagram to the right. If you'd like to fill it out before I go over the answers, feel free to hit pause. So if we look at the flow of blood, right, we've got blood flowing here. There's the capillary where filtrate's produced, and then you have a section where it's hard to say what we're doing without giving you the answer. So we'll just kind of go through, I guess. So at this first part, number 1, that's where the capillary is forming the filtrate. So blood is pumped through. Usually we have a larger diameter vessel here, smaller diameter vessel here, so there's a pressure change which pushes water and solutes through, and so that is filtration. The next step here, where we're reclaiming useful solutes, that is reabsorption. Here where we take things out of the blood, that is secretion, and then when it leaves the body, I guess I left the label in there, but that is excretion because it's exiting. Number 12, desert-dwelling mammals excrete very hyperosmotic urine, so very concentrated urine. What would you predict about the length of their loops of Henle, or the length of their nephron loop? They should have a very long nephron loop in comparison to other mammals. And then alcohol inhibits pituitary secretion of antidiuretic hormone. How would this affect urine production? So antidiuretic. So a diuretic makes you pee, okay? But antidiuretic is a not pee. So antidiuretic hormone makes you not pee. But if we inhibit the thing that makes you not pee, that would make you pee. So alcohol increases urine production, which some of you may know from experience. So we made it to the end. We talked about homeostasis, which is that stable state. We talked about the different kinds of animal tissues, broadly organ systems. We'll get more into detail on organ systems later. We talked about endocrine system, which is hormonal, and nervous system, which sends electrical impulses. And we talked about negative and positive feedback cycles and kind of the components of the negative feedback cycle, endotherm and ectotherm, so maintains your body temperature through metabolism or behavior. And then we talked about osmoregulation and different excretory systems. I would make sure to focus specifically on the kidney and how it works in more detail than the others, but be aware of the other excretory systems. So that is it, and as always, if you have questions, feel free to send me an email. Thank you.